Okay, so to review, there are intrinsic and intrinsic pathways to promote apoptosis. The extrinsic one involves either a soluble signal like TNF alpha or the cell-cell interaction with the FAST receptor that lead more or less directly to the activation of a activator caspase and executioner caspase. Then there is a mitochondrial or intrinsic pathway that involves what is essentially a balance between pro and anti-survival members of this BCL2 family. So you can think of survival versus death as a seesaw between those family members. And if one type, pro or anti-survival, exceeds the other, then that's the fate of the cell. Is another example of how cell biology works. You have these yin and yang forces, and it's the relative concentrations of those two oppositely oriented, often protein components that ultimately dictate the cell fate. There's a very good reason why that is. I'm just not going to have time to go into it. But anyway, there's one additional component to this that is also yin and yang, and that is a protein that I'm not sure is identified on any of these slides, but it's called inhibitor of apoptosis proteases, IAP, that even in the presence of active executioner caspases can shut down the decision to undergo programmed cell death. So they're sort of like the last defense against accidental apoptosis. And it turns out that they themselves are inhibited because there are components that are released by the mitochondria that inhibit those and therefore tip the scales towards caspase 3. And those things are exemplified here by this XIAP shown here. SMAC Diablo is one example of that protein. And we'll see that in the next slide, which is a list of some of the 1,000 or so substrates that are proteolytically targeted, mostly inactivated. Sometimes they lead to an activation event as a result of the proteolysis. So some of these are just part of the apoptotic regulatory pathway itself, so we just covered all of these. Remember APC of the beta-catenin story. Also, beta-catenin itself is a target, E-cadherin. So basically all of the signaling and response from the Wnt pathway and from attachment, all attachment-mediated signals are disposed of. And there's focal adhesion kinase, so the integrin-based signaling is gone. That blebbing that we talked about, part of that is through specific targeting of cytoskeletal elements, and all of these are those. Plectin we talked about a long time ago with its association with protein kinase C, so it's the target. These are nuclear cytoskeletal elements. Nuclear lamins underlie the nuclear membrane. So if they get proteolytically cleaved, then the nucleus fasciculates. That happens actually during mitosis, but it happens as part of apoptosis as well. And here's cyclins. Inhibitors of cyclins, the retinoblastoma protein, from the cell cycle. ICAD is that inhibitor of the caspase-associated DNase that leads to the fragmentation of DNA. We never actually talked about PARP. NF-kappa B, remember that from way back when. There's our old friend AKT, calmodulin-dependent kinase, the MAP kinase, MAP2K, protein kinase C, RAF from the MAP kinase pathway, RAS, GAP from receptor tyrosine kinases, phosphodiesterases from G protein coupled receptor responses, and HSP90. So it's a very red carpet list of proteins that are targets of caspase, and some of those that make sense in terms of the response that you see, why they would be targets. And then finally, I haven't talked about coronavirus for a while, so I wanted to end the class basically with what is known about the relationship between coronavirus infections and some of the things that we talked about towards the end of the class, there really isn't much in terms of signaling that coronaviruses seem to do. But there does seem to be a growing list of associations between coronavirus proteins and the regulation, generally the stimulation of apoptosis. And this is taken from a recent review that actually is not so recent that it includes SARS-CoV-2. It's really more about SARS. But what seems to be clear is that there is a collection of proteins, particularly the envelope proteins of SARS, that among the things they do is to promote apoptosis. In some cases, no one knows how, but in some cases there seems to be an inhibition of the standard pro-survival proteins, so a very classical way of triggering apoptosis by inhibiting the inhibitor of apoptosis that's shown here. Actually, I didn't even talk about TBID, which is a way of linking the extrinsic and extrinsic pathway. But in any case, there is some evidence that SARS proteins, among the things that they do associated with the viral infection, is triggering apoptosis. So I did want you to be aware of that. And that's that.